Hi, and I'm so pleased to be here today to speak to you about the vaginal microbiome and susceptibility to urogenital infections. These are my disclosures and acknowledgements, and I did want to take a moment to pause and say that we're discussing the vaginal microbiome today, but of course, not all people with vaginas and hence a vaginal microbiome will identify as women. And I'll try to be as mindful as possible of that. But if I use cis women-centric language, it's either for clarity, for ease of discussion, or because of the language used in the research cited, and truly it is not intended as an erasure for other important demographics. So our objectives by the end of this presentation, you'll be able to identify the basic elements of the vaginal microbiota, identify some ways in which possibly the vaginal microbiota may protect against acquisition of sexually transmitted and urogenital infections in women, and identify some challenges in developing novel microbiome-based improve women's reproductive health. This is going to be sort of a very broad overview talk, just touching lightly on some of these subjects, but I hope you'll find it interesting. So today we're going to talk about the vaginal microbiome, and so discuss a little bit about what we mean by that. So when I talk today about the vaginal microbiome or microbiota, I really mean the communities of bacteria inhabiting the vagina. Now, as you know, our bodies are covered with bacteria. There's specialized communities living in each anatomic area that are adapted specially for that niche, and the vagina is no exception. I think it's really remarkable that in the last 20 years or so, we've realized that some of these bacteria that are living on us can cause harm and can cause disease, but others may actually be critical to our health. A really interesting concept, and one that definitely applies to the vaginal microbiome. Now, I'll pause and just say that, of course, technically, microbiota refers to the microbial species present in a community, whereas microbiome refers to the microbial gene repertoire. But I'll use those interchangeably today, again, really to reference the communities of bacteria, in this case, inhabiting the vagina. Now, another note, we're going to be focusing on the bacteria today because that's really where most of the research has occurred. But keep in mind, they're not the only ones that are there. There is a virome, a microbiome, so fungi that might be present in that location, even a parasitome, and those are very important emerging areas of research. But as I said, most of the work to date has been done on bacteria, and that'll be our focus today. So what is the importance of the vaginal microbiota or microbiome? Well, we think it's very important because in the healthy or optimal state, we think of the vaginal microbiota as the body's first line of defense against invading pathogens. And so an optimal or healthy vaginal microbiome, we think is dominated by a few Lactobacillus species, which, as their name suggests, produce something called lactic acid that acidifies the vaginal pH and is very protective against things that might like to invade, like gonorrhea or chlamydia or even E. coli. This is an image up here to the right of these nice long rods, lactobacilli, on top of a vaginal epithelial cell. And there are a number of mechanisms by which lactobacilli may act to prevent infection with some of these pathogens that we'll discuss later in the talk. Now, in some women, for reasons that are incompletely understood, those vaginal lactobacilli are decreased, and instead there's an overgrowth of a variety of gram-negatives and anaerobes, things like Gardnerella vaginalis, Prevotella, Mobiluncus, Smithia, Adipobium, etc. And these individuals have lower lactic acid in the vagina and an elevated vaginal pH. And that condition, that low lactobacillus condition, is called bacterial vaginosis, or BV. Now, about 50% of women with this low lactobacillus state, this bacterial vaginosis, are symptomatic. And symptomatic BV is, in fact, the leading cause of vaginal complaints in women of reproductive age in the United States. Women classically complain of vaginal odor or discharge, though they may have some other symptoms. And clinically, BV is diagnosed via the Amsel's criteria. So if a woman comes into a doctor complaining of things like vaginal odor or discharge, the doctor will do a pelvic exam, look at the vagina, and diagnose her with BV if she meets 3 out of the 4 Amsel's criteria. And those are having a thin, homogeneous vaginal discharge, so a characteristic discharge, an elevated vaginal pH, clue cells, which are vaginal epithelial cells that are covered by a mat of BV-associated bacteria. And you can see a clue cell on top of a normal vaginal epithelial cell in the image here on the slide. And then having a positive amine or whiff test where essentially KOH is added to vaginal secretions and produces a fishy or mean odor. And so clinically in symptomatic women, BV is diagnosed via the Amsels criteria. For research purposes, for many years, we've used something called the Nugent score, which essentially quantifies different morphologies of bacteria present on a Gram stain and essentially estimates what proportion of those are those healthy, optimal lactobacilli as compared to the BV-associated bacteria. More recently, there have been some molecular tests that have become available, including some microbiome-based tests that essentially quantify, based on sequencing, how much lactobacilli, as compared to some of these BV-associated bacteria, are present in the vaginal microbiome. Now, I think something that's really interesting about bacterial vaginosis, or BV, is that it really is, we think, a disorder of the vaginal microbiota. And if you think about it, we hear a lot about the microbiome, but there are very few diseases that we can identify as a disorder or dysbiosis or messing up of the microbiome. Perhaps you could say that C. diff diarrhea is one of those, but this is really the most clear example I can think of. And I think as a prototype entity, it's very interesting from that perspective. Now, in the last 15 years or so, with the advent of new molecular techniques, things like 16S RNA gene amplicon sequencing, metagenomics, et cetera, we've really developed a more in-depth knowledge about the vaginal microbiome and bacterial vaginosis. And many of the bacteria that are present in the vaginal microbiome are anaerobes, so they're very difficult to culture. These sequencing techniques have really enabled us to identify the full depth and breadth and complexity of these communities that inhabit the vagina. So what we know now from these new techniques is that the vaginal microbiome of at least reproductive age women can be categorized into a number of different community to state types, or CSTs, based on composition and relative abundance. And 4 of those are dominated by lactobacilli of different kinds. The last, or CST4, is a low lactobacillus, essentially BV-like state that's dominated by a variety of BV-associated bacteria. And enabling this sort of more specific characterization of the microbiome has really enabled us to make some to have some important insights as well. I think the first of those is that bacterial vaginosis is not monolithic. It's not that all women with BV have exactly the same vaginal microbiome. So CST4, that BV-like state, is commonly grouped at least into 2 and sometimes 3 different subcategories, depending upon which bacteria are there. So it's not that all women with BV have exactly the same microbiome. And that might be important in the long term in thinking about how we treat BV. Now, in terms of other important insights, we've recently, with these new molecular techniques, been able to identify lactobacilli down to the species level. And so I think that's important because what it's allowed us to do is then relate those species level associations to outcomes, such as an increased risk of acquisition of STIs. And our old paradigm was essentially that lactobacilli are good and BV-associated bacteria are bad, because lactobacilli are associated with protection from STIs and other bad outcomes, whereas BV is associated with an increased risk of acquisition of STIs and other infections. But I think with this new identification, now we've started to make these associations with more nuance. And what we're starting to see is not all lactobacilli are equally protective. So we'll talk about this more in a little while, but Lactobacillus inners in particular appears to be less protective than Lactobacillus crispatus in protecting against acquisition of STIs. Now, the vaginal microbiome differs throughout life, and there are some important things that influence the vaginal microbiome. One that I wanted to highlight is the role of hormones. So before menarche, before girls have much in the way of estrogen, lactobacilli are low. But as women go through menarche and are in their reproductive years, estrogen levels are high and lactobacilli increase as well. So estrogen really supports the growth of lactobacilli in the vaginal microbiome. Then in menopause, levels of estrogen decrease and lactobacilli decrease as well. Even within a menstrual cycle, the hormones can influence the vaginal microbiome. So for instance, this is a study which shows that when levels of estrogen are high in the red line here in the middle of the cycle, the vaginal microbiome is more stable than at times when estrogen is low, and particularly more stable than at times of menses, where we think that blood in the vaginal vault also acts to destabilize the vaginal microbiome. Now, I talked a little bit about this question of whether all lactobacilli are created equal and what their protective properties may be against STIs. And we think a lot of that might have to do with the lactic acid that they produce. So Lactobacillus crispatus dominated vaginal microbiome tend to produce a lot of lactic acid and have a very low pH. So these green dots here are vaginal microbiota dominated by Lactobacillus crispatus, and they're colored green because the vaginal pH is very low, less than 4.5. Lactobacillus inners dominated vaginal microbiota, on the other hand, sometimes have a low pH, but sometimes have a much higher pH. And we know that Lactobacillus inners is not as good at producing lactic acid. Then that diverse BV group in the middle there, CST4, tends to have very high pH because there's very little lactic acid. But the story may be even a little more complicated than that. It may not just be lactic acid, but it may in fact be particular isomers of lactic acid that are important. And so there are two isomers of lactic acid, D-lactate and L-lactate, that are produced by some lactobacilli. Lactobacillus crispatus here you can see is very good at producing D-lactate in the red as well as L-lactate in the blue. Whereas Lactobacillus inners essentially doesn't produce D-lactate at all, though it can produce some L-lactate. So keep that in mind as we talk about mechanisms later on in the presentation. Now we'll talk about a number of different STIs, including chlamydia and gonorrhea, which are the number 1 and number 2 most commonly reported STIs in the United States. These are very common, unfortunately, particularly in younger women. They're usually asymptomatic, but women can have vaginal discharge, pain with urination, or nonspecific symptoms. They can cause a cervicitis or infection of the cervix. And this picture at the top right here is a very infected, inflamed cervix that's red and has pus, essentially. And untreated, both gonorrhea and chlamydia can lead to pelvic inflammatory disease, infertility, and chronic pelvic pain. Trichomonas is another important STI. It's actually the most common non-viral STI in the United States. It's caused by a parasite. It's commonly asymptomatic, but it can cause a vaginal infection with vaginal itching, irritation, pain with urination, discharge, and odor. And classically, on exam, a clinician can see what's called a strawberry cervix, which is a cervix here in the picture at the lower right-hand corner of the slide that has these punctate red hemorrhagic areas. Another STI we'll discuss in the upcoming slides is human papillomavirus. That is HPV, which causes it causes cervical cancer as well as genital warts. And that is actually the most prevalent STI in the United States. So let's talk about some of the associations that really is the focus of this talk. So this is a meta-analysis where a group looked at associations in a number of different studies between having a low lactobacillus vaginal microbiota, i.e. that BV-like or dysbiotic state, with having a gonorrhea infection, whether incident or prevalent. And they found a trend towards an association between an increased risk of gonorrhea if you had a low lactobacillus or BV-like state. I'll say that there have been a couple of large epidemiologic studies that came out since this one since this meta-analysis was done, that showed even more strongly that there is a link between having a low lactobacillus vaginal microbiota, i.e. BV, and having an increased risk of incident gonococcal infection. So really this risk of gonorrhea acquisition with having a dysbiotic vaginal microbiome. Similarly, with HPV, human papillomavirus, which, as we mentioned, causes cervical cancer as well as genital warts, there is an increased risk of prevalent HPV infection in those who have BV as compared with the lactobacillus-dominated vaginal microbiota, as well as an increased risk of the HPV persisting and not going away, not being cleared. And that's important because persistent HPV is more likely to lead to cervical cancer. Very similar association we see between having a low lactobacillus vaginal microbiota and increased risk of incidence and prevalent trichomonas infection, that parasitic infection we talked about. And lastly, there's a strong association between having a low lactobacillus vaginal microbiota, i.e. BV, and having incident or prevalent chlamydia infection. Now, I mentioned that we certainly see this association between having a BV-like state and an increased risk of acquiring chlamydia. But what about L. inners? I had started to talk about we're having some indications that that one may not be as protective as some of the other lactobacilli. And so there were a couple of studies, one done by Vanderveer, which came out in 2017, and one done by Van Hoot, which came out in 2018, which looked at the risk of chlamydia acquisition in those who had a low lactobacillus state, an L. crispatus dominated state, and an L. inners dominated state. And so Vanderveer, for example, they were looking at women who were contacts to individuals diagnosed with chlamydia and looking to see who actually acquired it, who tested positive for chlamydia. And they found that those who had a low lactobacillus or BV-like vaginal microbiome were at an increased risk of chlamydia as compared to those who had a lactobacillus crispatus dominated state with an odds ratio of about 4.2. But intriguingly, they also found that those who had L. inners were at an increased risk as compared to those who had L. crispatus dominating their vaginal microbiome for chlamydia infection with an odds ratio of about 4.4. So very interesting, and those results were similar in the Van Hoot study, showing that we see an association between an increased risk of acquiring many of these STIs and having a low lactobacillus state, but also lactobacillus inners in particular may not be as protective as lactobacillus crispatus. What are the mechanisms here? Well, this is an area which I think it needs a lot more research. This is all very preliminary, but we do think that lactic acid may play an important role. So there are a number of studies, including one I'll show you on the next slide, which suggest that at least in vitro, lactic acid can inhibit the growth of things like gonorrhea or chlamydia or their infectivity, and that low pH is critical here too. Also, lactobacilli may bind very tightly to vaginal epithelial cells, and through competitive binding, prevent the binding of things like trichomonas or gonorrhea, and prevent them from establishing a foothold in the vagina and causing infection. Immunomodulation might be important. We know that lactobacilli tend to tamp down inflammation in the vaginal microenvironment, and perhaps that results in better barrier epithelial integrity that prevents infection. We don't know for sure, though. And then there are metabolites other than lactic acid that may be important as well. So Chlamydia trachomatis requires tryptophan for its growth, and it has to acquire that from the host cell. So one of the key defenses against chlamydia is that the host cell is capable of depleting tryptophan and essentially starving chlamydia of this very important factor. But in vitro studies, at least, have shown that certain BV-associated bacteria, certain strains of Prevotella in particular, can produce a substance called indole that chlamydia is capable of turning back into tryptophan and can actually rescue the growth of chlamydia and promote chlamydia persistence. So perhaps some metabolites produced by BV-associated bacteria may actually enhance susceptibility to or persistence of infection. This is another study that I mentioned looking at the role of lactic acid, in this case for chlamydia. They did in vitro Chlamydia trachomatis secondary infection assays, and they essentially incubated cells with the supernatants from cultures of Lactobacillus crispatus in the orange on the right here, Lactobacillus jenseni in the yellow, and Lactobacillus inners in the light orange, all here on the right. And then did this chlamydia infectivity assay at 0 hours, 24 hours, and 48 hours. And what they found that there was significant inhibition of chlamydia infectivity in those experiments where the cells had been incubated with a supernatant from Lactobacillus crispatus and Jensenii, that effect was definitely not as strong with Lactobacillus inners. And then they tried to tease out whether that was due to lactic acid or not. And so they did similar assays where they incubated the cells either with a control in the light gray or with D-lactic acid in the dark blue, L-lactic acid in the pink, or HCl, so just hydrochloric acid, to see if it's just a pH effect in the purple. And then they did the assays again at 0, 24, and 48 hours. And what they found was that there was definitely increased inhibition of chlamydia trachomatis infectivity over the control when using D-lactate, to a lesser extent L-lactate, and really almost not at all with HCl, though that effect was somewhat attenuated with a longer time, particularly in the 48-hour time frame. So lactic acid seems to be very important here. HIV, we've actually known for a long time that BV is associated with an increased risk of HIV acquisition. This is a meta-analysis done as far back as 2008, which showed that women with BV had an increased risk of both incident and prevalent HIV. Some of those studies have now begun to be replicated in the era of new molecular assays. And so this was a study done by Gossman where they followed a very high-risk cohort of women in South Africa to see which women acquired HIV and which did not, and tracked their vaginal microbiota, tried to relate it to that HIV acquisition outcome. And what they saw was that women with a BV-like state, so low in lactobacillus, in this case, they said their community state types were CT3 and 4, had an increased risk of acquiring HIV, a significantly increased risk of acquiring HIV over those who had a Lactobacillus crispatus dominated state. They did find that some women with Lactobacillus inners did acquire HIV, though at a lower rate than those who had BV. And remarkably, not a single woman with Lactobacillus crispatus dominated vaginal microbiota in this entire study acquired HIV. So really an amazing sort of protective effect that was seen there. What are the mechanisms? Again, these are under study, but lactic acid certainly in vitro has been shown to inhibit HIV activity. And we think that inflammation may be very important here as well. So activated CD4 T cells that express the HIV co-receptor CCR5 are more prone to infection. And there are certain inflammatory or pro-inflammatory cytokines and chemokines in the vaginal microenvironment that have been associated with HIV acquisition, things like IL-1 alpha, TNF alpha, etc. And again, the Gossman paper showed very clearly that women who had a BV-like state, that CT3 and 4, had increased levels in the vagina of certain pro-inflammatory cytokines and chemokines, things like IL-1 alpha, TNF alpha, and some others that have been associated with HIV acquisition. So maybe inflammation is very important in mediating this relationship with HIV. Lastly, just to end, what about urinary tract infections in women? Well, I would say this is much less studied. The association with the vaginal microbiome and susceptibility is much less studied. But it is an interesting area of emerging research. And urinary tract infections are certainly an enormous problem in women. They are the most common bacterial infection, and postmenopausal women in particular are disproportionately impacted, many of them experiencing chronic recurrent UTIs, which can really be devastating. Now, UTI pathogenesis, classically in women, we think of E. coli coming from a gut reservoir to colonize the vagina, and then under the right circumstances, ascending from the vagina into the bladder to cause a urinary tract infection. There are a lot of other complicated mechanisms here, but that's the basic idea. And so the vaginal microbiome might be very important in determining that intermediate step in the middle here, whether E. coli is capable of colonizing the vagina, because that's a key step in the pathway ultimately leading to urinary tract infection. Now, there have been some cohort epidemiologic studies that suggest that women with vaginal lactobacilli are less likely to have introital colonization with E. coli and less likely to have an E. coli UTI. That was shown by Hooten back in 1989. And then an interesting study done by Raz in the New England Journal in 1993 randomized postmenopausal women with recurrent UTIs to either get intravaginal estrogen or placebo. And interestingly, they found that women who got the intravaginal estrogen were both more likely to have lactobacilli detected after the intravaginal estrogen in the vaginal swabs, and also less likely to have UTIs after the intravaginal estrogen. So perhaps in this case, the promotion of vaginal estrogen led to increased lactobacilli, which perhaps led to some protection against UTIs. But that's really an area that needs a lot more research. There is some suggestion that perhaps competitive binding, just as with STIs, could be important in preventing E. coli from binding in the vagina. And also there are some in vitro studies that I won't discuss in too much detail, but there's one by Garty et al. Here that show that in vitro E. coli seem to be inhibited by lactic acid and by the supernatants produced by some lactobacilli. So really, I think this brings up the question. We've seen now that there are these associations between having a low lactobacillus vaginal microbiome, or BV-like state, and an increased risk of gonorrhea, chlamydia, trichomonas, HPV, HIV, and perhaps even urinary tract infections, though again, that's an area of research that needs more work. But I think there are a lot of challenges here. I think it really raises the question, could we actually optimize the vaginal microbiome to prevent these infections and improve women's health? This is a really exciting idea. But again, there are a lot of very important challenges along the way. Number one is that bacterial vaginosis is very, very difficult to treat. Nearly 30% recur within 1 month and 60% within 12 months after antibiotics. Part of that may be that our tools at present are very blunt. So really all we have to give is antibiotics, and we essentially blast the vaginal microbiome, knock down the levels of BV-associated bacteria. But really, I think if you think about it, we'd want a way to put back some of the healthy and protective lactobacilli as well. We don't have a great way to do that yet, at least not with our commercially available treatments. Additionally, hormonal issues may be a complicating factor, particularly in postmenopausal women. So if you just don't have the estrogen there to support the growth of lactobacilli, perhaps just treating with antibiotics isn't going to do much. So perhaps you need to supplement with other things like that intravaginal estrogen, or maybe there are other factors to consider. And then there's another very important issue, the contribution of sex partners. So before women become sexually active, they don't really get bacterial vaginosis. Studies treating male partners to try to reduce recurrent BV in the female have not really resulted in an improvement, but there is this question of whether there's an element of reinfection associated with a sex partner. And so that may be a complicating factor when we're trying to figure out how to optimize the vaginal microbiome, treat bacterial vaginosis, and put women into a lasting, stable lactobacillus-dominated state. There have been some exciting developments recently, however. There are a number of things to consider, but one I wanted to highlight was a high-profile paper that came out in the New England Journal by Cohen, where they essentially randomized women with BV to, after getting treated with antibiotics, getting an intravaginal probiotic of Lactobacillus crispatus versus a placebo, and they treated them for 12 weeks. And at 12 weeks, they saw that in the Lactobacillus crispatus probiotic group, there was a 30% recurrence, whereas there was a 45% recurrence of BV in the placebo group. And that was statistically significant, though that by 24 weeks, it was not so significant. So I think only in the sort of terrible world of recurrent BV would we be excited about a 30% recurrence rate just at 12 weeks. But I think it is really very exciting and interesting because it shows that as a paradigm, perhaps we can actually optimize the vaginal microbiome, treat BV, and then perhaps in the long term, improve some of these outcomes and protect against urogenital infections in women. So again, many challenges. I think another big challenge is really that BV treatment, it's not clear at this point exactly if treating with antibiotics is optimal, some women may have increases in cytokines and inflammation in the vaginal microbiome environment. There was at least one study that suggested that, though I think more research is needed. And then lastly, what is the right probiotic for the right woman? Is that really the same for every woman? Why should we think, for example, that a probiotic strain of Lactobacillus crispatus developed and isolated from a woman in the United States should be the same strain that's optimal for a woman in sub-Saharan Africa, for example? I think we need a lot more research to really understand what is the right probiotic, or what is the right bacterial strain, what is the right vaginal microbiome that's optimal for the right woman. But I think, again, this really raises a question of the promise of the future, and that is really that perhaps we'll be able to develop vaginal microbiome-based therapeutics that can prevent urogenital infections in women, and I think that's a very exciting prospect. So I'll end there, and I did want to say some thank yous to Rebecca Brotman, Khalil Ghanem, Jacques Ravel, and Victoria Handa. Thank you.